This talk is about the enhancement of gene regulatory network inference using an approach based on Markov Random Fields to integrate different heterogeneous data sets. And the talk was first given in a shortened version at last year's Dream Recon Conference 2018. And so the motivation for this work that was done at the Carnegie Institution at Stanford is that nowadays the reverse engineering of gene regulatory networks does remain a challenge, in particular for eukaryotes, because first of what is known as the curse of dimensionality problem, which is that we often have large number of genes, but only small set of gene expression data sets, or say, particularly if you're looking into condition-specific network inference, then we're given small sets of conditions versus large number of genes. And what might be even more important is that within eukaryotes, the gene expression levels, the regulation levels, are further affected by other aspects such as chromatin modeling, other processes, post-transcriptional or post-translational processes and mechanisms. And these are not captured within the gene expression data sets. And then finally, we often only have like small gold standard data sets, and within these data sets being defined as experimentally validated cases of regulation between transcription factors and target genes, what we lack is oftentimes true negative, that is experimentally validated cases of non-regulation. So the idea being that we try to include as much data sets as we find that would make sense to corroborate found networks or prioritize links. But what we have to take into account is that oftentimes these data sets only are available partially. That means that they do not cover the entire genome, for example, and so therefore we have to make sure that we integrate these data sets in a non-biased manner, in the sense that we do not prioritize some stuff that we already know and neglect other potentially interesting links where we didn't have the additional data sets for. And that is where the idea of using Markov Random Fields might help. So going one step back, what is a Markov Random Field? So traditionally, the idea has been applied a lot in computer vision before deep learning came around, particularly. Here, what you can see in computer vision in scene recognition, object classification. So here's given an example. What you see here is the idea of recognizing man-made structures within natural scenes. So what you see on the right actually is we have the image divided as a grid cell based layer, and within each of those grid cells we classify, in this case based on certain handcrafted features that are supposed to represent man-made or natural areas, and we do a classification per grid and then assign a square in case that we have predicted man-made structures. So as you can see on the right, what is correctly classified is this house in the middle of the image, but what is faultily classified is the one on the right. And what we can now do to improve this is to use this idea of checking the surroundings around each grid cell. So, and this is particularly done on the left with this approach called dual support vector fields, which is a variation of the Markov Random Field. So what we do here is that we check what is the strength of the classifier, what is the prediction confidence, so to speak, of the classifier per grid cell, and then looking at its surroundings. So we can see here that the grid confidence, the prediction confidence for a grid cell for this man-made structure in the middle tended to be very high. So even though the surrounding areas were predicted to be natural, this wouldn't have any effect on the grid cell predictions for this man-made structure in the middle. Whereas on the right, we could see that the surrounding predictions of natural area had been so strong in contrast to apparently a very weak confidence prediction for man-made structures within the grass, so that this has been basically flipped, and the predictions of the surrounding area has been chosen to be the more appropriate classification for this area. So the idea basically being, and here we call these grid cell superpixels, it's just basically a cluster of pixels. So we include neighborhood information. And so this is basically the idea of Markov random fields and variations. And this could now be applied, or this is something that we now apply to our gene regulatory networks enhancement problem. So, and there we design the Grace algorithm, which is supposed to do this. So initially, what we start with, and this can be, it's kind of a modular three-step algorithm. So the individual modules, in particular module A, can be switched for something else, which is the idea that we start with building an initial gene regulatory network. And here what we do is we build this from expression data and filter it by promoter binding in order to get direct links for making the network more accurate. And therefore we derive, we set some threshold, and again this is something that can be based on users' preferences or based on a probabilistic model, in terms of saying, okay, when does our inference strength between a transcription factor and target go below a certain value that would be expected by chance given the expression data set. So there's different ways here, but so we end up with an initial gene regulatory network, and the next step now being that we look at individual small modules of co-regulation. So this is really diving into the network, and what you can see here is to say, okay, the red dots representing transcription factors and the yellow dots representing targets. In this case, what you can see, the initial network sort of made an inference that this transcription factor is acting on both these targets. Might be more, but here we have this example of 2, because what we want to show now is that we basically change our perspective on this initial gene regulatory network. So what we're doing is we transform the network into what we call a meta network, and that is that we now, from having the genes as nodes and the links as the regulatory links that have been inferred, we switch to now the nodes being these original links. So in this case, in our example, where we started with one transcription factor having 2 links to the 2 targets, we switch this into 2 nodes that represent the 2 links, and the link between those is basically the interaction strength based on some additional metric and data set between these targets. So this is to support this idea of the transcription factor co-regulating these target genes, because we assume that within additional data sets this might show up that these target genes do have something else in common. And again, this could be different data sets, and we're going to look into this in a second, but this could be different data sets that we might use here. And so what the intention is that we then say, okay, each link comes with a confidence value. And so since we've been using an initial threshold, these 2 links already should have a decent confidence, but now we can use this to prioritize or even further filter the network. And the idea being that we say, okay, if one of those links has a very high confidence, the other one potentially not as high, if we find this relationship between these targets to build on this idea of co-regulation, then we might say, okay, in this case, even the regulatory link that doesn't have that high a confidence is kept because it's sort of related to this target, and that target does have a very much more confidence of being regulated by this transcription factor. And so we kind of produce this sort of triangle, this relationship between a transcription factor and two targets, and the relationship between those two targets among themselves. And now what we do, where this idea of Markov Random Fields comes in as sort of the technical underpinning, the statistical framework to solve these optimization problems that we generate, is that we first extract all these individual modules. And in this particular design, and this design of Grace, it's basically a transcription factor and several other potentially co-regulated target cut out of the original network. And what we then do is we get modules, individual modules, and we apply Markov Random Fields to each module. And what combines all these modules in order to make also to have this model not to be completely arbitrary is that the parameters are shared. So we're going to look into this in a second, but this is actually a crucial aspect. Otherwise, we couldn't really control that the model converges to something reasonable. So we do apply the same Markov Random Field structure to each module. But what we also do is that all the modules share the same two model parameters, and that's the way that we can train them and optimize them with a limited gold standard data. So, as I mentioned, what we need to reflect the co-regulation between two target genes is a given metric. So what we define here is a pairwise distant metric, and this is defined as follows. First of all, we have the distance in the, what you can see here is the confidence levels for the transcription factor to regulate each target, each of the two targets. This is given by this value. And what we first do is to build the difference to see, okay, how much does this confidence level differ between the regulation of this transcription vector to target 1, target 2. And obviously what we want to have is actually pretty small difference. That would be favorable in terms of assuming co-regulation. The second connection is what I mentioned based on this additional data sets to link the two target genes in this transformed network view. And this comes from an additional data set. This could be correlation between the targets, or other data sets such as protein-protein interaction. So here we would prefer a high value. The total value is sort of normalized, so as you can see, as I mentioned, we want to have a small difference between the regulatory values, so this is below the denominator, and also we want to have a high value with respect to the co-regulation. So what we want to have is a high value in, say, PPI or co-expression between the target genes. So this goes on top, and this is how we generate this distance metric in order to reflect this co-regulation between the two genes. And that's actually how in this meta-network the links are defined. So we have the nodes and the links, and now we can express this in our Markov Random model. So first, How is it defined? It's basically this probability distribution. This goes with an exponential, an energy function as an exponential divided by what's called a partition function set in order to normalize the whole formula to provide a probability distribution. And this is interesting given that this sort of formulation allows for the energy function to basically take any form as long as it's positive. And given the partition function and the definition of the partition function set, we will get a valid probability distribution. So now the interesting part being the customizable energy function, the cost function. And this is basically defined for a single Markov random field model. What you see here is, first of all, we have no potential, and then we have pairwise potentials. This is sort of how the definition of Markov random field goes. And the node potential is actually the value at a certain node, L. And we build a sum over all these nodes, and then the pairwise potential is concerning the links between each nodes. So in our example, going back one step, to make this very simple, on the right we would have a very basically sort of the simplest Markov random field model that we could have. What we have here is the blue nodes would be the node potentials, and then the link between is the pairwise potential. So how this is defined is that for each node, what we have is the original confidence value for these regulatory links that make up the node potentials, and then between those nodes, what we have is the pairwise potential, and this is defined as this correlation metric. So the idea being that we want to see if you will, maybe we could visualize this in a way that we say, okay, whenever we have, say on the left, we have a very high confidence value assigned to this node as being the node potential, then if this pairwise potential, this link between these two nodes is high, then this sort of flows across this link onto the right node, and therefore helps this right node to to be also even if the node potential on the right is a bit lower, it's sort of the connection between the two leads the model to classify, to still keep this right node as a valid link in our case. If we think back in this idea of in the man-made structure recognition, we sort of had the same, where we had this problem of having these connections between grid cells and having a strong man-made scene and a not as strong natural scene, and then basically we would flip the sign. So here we kind of do the same, just to keep in mind that how we transfer this problem. So I'm picking this up again. Maybe I'm going to pick this up later as well. But this is, again, this is the mathematical formulation of this cost function. So we do a cost of all these node potentials, and again, here you can see, here you can, with this C, you can actually refer to these values as the link strength. In the left part of this equation, the sum over all these 3x. And then we add all these pairwise connections, pairwise potentials. And what we then basically can do is set this up in terms that the model can actually work with it. And here actually maybe this makes it a bit clearer that what we see now is so the XL represents is basically a binary variable. This is actually whether the model later it can only take 0 and 1, and actually it goes that we keep a certain node representing a certain link, or whether we want to neglect it. And then this is basically making use of this, of the confidence value for this link. And what we can see here now is that actually for the model we want to minimize, the idea is we have this duality here, and we want to minimize the cost functions. If you look in the equation of this probability distributions, minimizing the cost function is actually equivalent to maximizing the probability for the whole system. That's basically finding the most probable system given the data. So here, and this is maybe otherwise that might be a little bit confusing why the equation is set up this way. So we look at the node potential, the sum over the node potentials. This is the first part of the equation on the right side. As you can see, if we have a high value of v, if our confidence level for a certain link is high, then for a model it's favorable to set x to 1. Setting x to 1 instead of 0 would mean the model keeps this link, keeps this node, which is the link in the original initial network. And because why is this favorable? It's favorable because in this case 1 minus 1 goes to 0, and then this high value, this high v, this high value basically vanishes. So whenever, and that basically translated into more straightforward explanation, what that means is that, okay, if we have a high confidence for a certain link, then the model can use this trick to minimize the cost function by keeping this node. So as you can see, this might be a little bit unintuitive at this point, but that's actually how we can make use of this fact that we can basically design our cost function with the only constraint, or only main constraint, that the cost function being positive. And we will later get in the overall framework, we're going to get a valid probability distribution. So this is concerning the individual node potentials. Now, if we go and say, okay, now we want to analyze the links between these nodes, between our relational regulatory links, what we're going to do is we kind of do the same thing. So just taking this example we had here, where we have this most simplistic Markov random field we can think of, the two blue nodes connected with this dashed black line. We have looked at the two node potentials. Is the two blue dots. Now we're looking at this dashed line in between that would make up this pairwise potential in our model. And now the basic idea that is sort of transformed into mathematics here is just like on the right side of the equation, preceded by this lambda, and I will mention what that means in a second. But what we can see here is, again, we have this value of the relationship between the two target genes. That can be either low or high. The higher it is, the more interesting it is to have this connection between the two nodes. So how is this translated into mathematics? What we see here is what's most important is this xl minus xl dash. So what this basically says is, this is the labeling, right? So if we assign label 1 to xl, which basically means keeping this link, and assign x0 to l dash, that would mean neglect this link, then basically the difference, the absolute value of the difference being 1. If we assign the same value, difference being 0. So we want the model, first of all, we want a model favor same. But this is expressed as follows, that we can say, okay, if the two are different, we're going to get, as absolute value, we're going to get difference, we're going to get 1. So the idea being, okay, if this is different, we get a 1, then we have to multiply this integral cost function with this relationship value, v crn l dash. So if this value has been high, then we have to sum this high value into our overall cost function that we want to minimize. And this would not really be favorable for the cost function. And actually, intuitively, if we look at it, it wouldn't be as well. So the idea being, if this VCRN-LL dash is high, then we would assume a strong relationship between these target genes, and we want these two regulatory links to be analyzed together and see whether we can keep one of the links even if its confidence value is not as high. So in this case, if we turn this upside down for the mathematical formulation here, we want to do, in terms of minimizing the cost function, for the mathematical model, it's favorable to say, okay, if this VCRN is high, then we should assign the same labels. So in this case, XL dash should be either 1 or both 0. So in this case, that we get the absolute difference being 0 as well. In this case, we multiply 0 with this value, and it basically vanishes in the cost function. So lambda is actually just a factor that can be learned during training as well. That's what I mentioned. It's one of the model parameters that we later train across all of these models and share across all of these models, is like, what is the influence of these links? So we, first of all, like with any other classifier, we initially look at each individual link, and we're interested in the confidence score for each individual link between a transcription factor and target gene. Now that we've included this relationship, this passing of information through relationships of target genes, we can define how high should this influence be, how influential should be one regulatory information between one transcription factor to one target be to this relationship of this transcription factor to another target, based on the relationship of the targets among themselves. And that's what this lambda does. So it basically regulates the influence of this additional neighborhood information. So basically we completed this model with an additional ingredient, if you will, and that is basically throwing in an additional threshold on on our confidence level strength that we do not want to go below, in terms, because here what we have to, and there's actually where this approach differs a bit from the one that we had with this man-made structure recognition in natural scenes, is that we, whenever we find very confident links, so that would mean links with a very high node potential, we do not want to lose them, even though all the other links are low confidence. This would actually otherwise make our model not very useful, because just because one transcription factor is linked to a lot of other targets with relatively low confidence, we would neglect the ones that might be actually very interesting. So we basically go the other way around. We say, okay, whenever we find something that is above a certain threshold, that is also be learned as the second parameter, second model parameter. We definitely want to keep this, and even though there's a lot of other potential transcription factor links to other targets that are linked with our target, that sort of would like to drag us down, that we that we basically would still keep this node. And yeah, so this is actually a very important ingredient, because, if you will, in the original idea of using the Markov Random Field approach for this man-made structure recognition, we didn't really care which direction we were going. Here, again, whether we were going to man-made or natural, here what we want to make sure is that very high confidence links are not removed just because the number of lower confidence increases. So this is actually a crucial ingredient. So, as I mentioned, what we now be doing is that we are defining a Markov Random Field model for each of these modules. So just remember, a module was defined as a transcription factor being linked to a number of targets that had relationships among themselves based on additional data sets. So this is actually what we define as a model. So therefore, in our initial regulatory network, we can extract a series of those models. And we can train Markov random fields on each of these modules, again, sharing the parameters. And this is actually what we're going to do now. We're building a likelihood or a combined probability by basically summing over all the individual probabilities from our individual Markov random fields using the same parameters. So we have this labeling for all our nodes x, referring to our original regulatory links. We have our parameters and theta, and these parameters, as I mentioned, are these two model parameters that we define. And we can use this to combine them and then basically compute total likelihood. And then we basically add one more step, which is that we do this basically several times using, so in a sense, just to give you one more information on the training process, how do we train this? We use available experimental regulatory link data, which is of course only partially available for some transcription factors and targets. And we basically make sure that we have some sort of overlap with our predictions and this experimentally validated data. And therefore we use those, and that's why GRACE is actually a semi-supervised approach, because we have our entire network, we have some overlap with gold standard data, and now we basically try to optimize the parameters theta in order to best reflect this gold standards. And therefore we, while tuning the parameters, we tune them, of course, since we share the parameters, we tune them for all the other random fields, for all the other modules where we didn't have any overlap. And that's actually the learning step that happens. And in order to make this robust and not to sort of overtrain, overfit, or just memorize the data sets, we repeat this process by subsampling the experimental data and building multiple of these models, and then finally integrating those. So basically for each module we not just build one Markov random fields. This process is repeated and repeated and repeated, because in the global view we've used several times, we've built this global model that combines all the individual module random fields, and we've used different types of the regulatory gold standard set time and time again. So we can build this, and what we did here is simplest version, doing an unweighted ensemble model by basically combining all these modules and then building the mean out of the results. And then what we can do is basically saying, okay, whenever we find a certain link that has been predicted by the model to be in our final network, whenever this came up more than 50 times, so equivalent to a likely probability of 0.5, then we keep it. Whenever it's been predicted by half of the ensemble models, we can also use this. So we can use this as a threshold. We can also use this as to assign like a meta confidence level to our initial links. And it enhances the robustness of the models, and not just to overfit and memorize the initial gold standard training data. So we did some comparisons with other regulatory network methods that have been performing well on certain benchmarks, or that try to make use of using additional data. Sometimes kind of combine this differently, and how we chose those, or how they're different, we've laid this out in more detail in the paper. But this is actually what we've been doing. And and then what we could see that on different sorts of data sets, the GRACE algorithm performed very well. So for example, we did this analysis for Arabidopsis, and on different Arabidopsis thaliana datasets. And what we were seeing is that for the regulatory link-based dataset, we achieved the highest recovery rates. Also for evaluating between target genes, whenever the final network that we recovered, what we could see is that also this quality had increased. That would be the co-functional pairs, gene ontology, co-localized target gene pairs, or with respect to metabolic pathways. So that is all concerning the relationship between the target genes and our final network. It's the last 3 data sets that you can see here that we evaluated on. And then we use this to recover, as well as doing de novo predictions for phase cycle control mechanisms in developmental programs of Arabidopsis. And yeah, you can see this a little bit more discussed in the paper, but basically we found some well-known leaf control mechanisms and cell division, cell proliferations mechanisms, and a few more. And that was very interesting, since we not only recovered but also found a few additional ones and made some assumptions about them. And yeah, so this brings me to the end of this talk. And basically, just in order to summarize, what we did is, or the original idea was to basically use an approach from computer vision, or in order to have a non-biasing integration of partial data sets. And also, this is also laid out in the paper, I didn't touch on this today, is that we build a slightly different variation of an optimization scheme that includes not only the recovery of gene regulatory, experimentally validated gene regulatory links, as is often done, but also includes some co-functional gold standards into one particular metric that we optimize on. And then finally, the whole system is set up to be a robust ensemble model in a semi-supervised fashion. Yeah, and the idea is that, again, the main idea of the program, to being able to having a high confidence network, and then even going one step further in terms to further filtering, prioritizing for experimental testing, and so on. And yeah, so the software can be accessed at jumpdna.ai. Thank you.